And now we will talk about causative agent of candidiasis. Candidiasis or candidosis are diseases named caused by fungi of Candida genus. The genus Candida includes about 200 species. Clinically significant species are Candida albicans, very often isolated from patients with candidiasis, and Candida tropicalis. What are they by morphology? Morphology: fungi of the genus Candida are oval budding yeast cells that form pseudohypha and septate hypha. But they rhizophotta, the smear from Candida, really they have oval shape, irregular size, and they multiply quickly, and they sometimes still stay together, looking like hypha, but really it's not hypha. They are separated cells, but they have just one thin layer, and so they stay together. In this smear stained by gram staining method, it's possible to use gram staining, and fungi have a color of ginseng violet. It is possible to use another staining method like Zell-Nielsen staining method. They will get red color. Then Giemsa staining method, they will get blue color. So it depends on staining method. Cultivation. Candida are aerobes. They grow on suberos agar at temperature 27 degrees by Celsius, means temperature, room temperature. Colonies are convex, white, shiny, looking like drops of mayonnaise. S-type colonies, glistening, a little bit fatty, and grows in tissues as yeast and are able to develop pseudohypha. In this photo, there are colonies of fungi of Candida genus on Suberos agar. So S-type colonies, just molds develop R-type. So yeast-like fungi develop S-type colonies. Epidemiology. Really, fungi of this genus Candida are detected in nature. They are detected on plants and in the soil and on the leaves of trees, and at the same time they are detected sometimes in human bodies, so they belong to normal microbial flora of mammals and humans. But I told you that they are able to exist on skin, on mucosa, not inside the body, not in internal organs, of course. In healthy people, they are able to multiply just on mucosa membrane of oral cavity, on nose and on the skin, and sometimes on the mucosa of genital tract, just in a very, very small amount. Just if we will do microscopy of the specimen from oral cavity, healthy person, if we will see just a few cells through the ocular, it's normal. So just a few cells are available, but not all people have Candida. Just a small amount of healthy people have Candida. And how it is possible to get Candida? Really, fetus in mother's body is sterile, but during the delivery, due to contact with infected tissues of mother, if mother has Candida, like normal microbial flora on mucosa of genital tract, the newborn will infected by direct contact with the tissues of mother, and now newborn has Candida. And sometimes the Candida transmitted by breastfeeding. So, and this Candida, if they have opportunity to multiply, they will multiply and may cause endogenous infection in patients with weakened immune system, in immune compromised people. Of course, HIV patients, the last stage of HIV infection is acquired immunodeficiency syndrome. Immune system is destroyed completely, and very often they die of candidiasis. Different clinical form, and they die on the last stage of HIV infection, of opportunistic infections caused by conditional pathogenic microbes, including Candida, including herpes viruses, and other, and pneumocystis very often. So transmission. Between the adults, Candida transmitted by sexual contact. And in this case, they cause orogenital candidiasis. And less common, transmitted by household contact to children at birth and during breastfeeding. Pathogenesis. The development of candidiasis is facilitated by the use of antibiotics for treatment of bacterial infections. On this, due to treatment of antibiotics, the amount of bacteria will be decreased, so they are not. And before fungi was multiplication of fungi were suppressed by bacteria, but if the amount of bacteria decreased, Candida are able to multiply. They will multiply on that. So the side effect of antibiotics was detected when antibiotics were produced. It was like miracle in medicine. It was possible to save the life of patient that should die of pneumonia, for example, and by treatment of antibiotics, the patient still survive, but finally, again, the person will die, and in post-mortal diagnosis, it was detected that he did die of candidiasis. Candida did multiply a lot, in children very often, and so we did realize that the side effects of treatment of antibiotics, and then we did detect other side effects of this therapy by antibiotics. Metabolic and hormonal disorders, immunodeficiencies, like diabetes mellitus, for example, leads to candidiasis. Increased skin moisture in countries with hot climate, Candida are able to multiply on the skin, and damage to the skin and mucosa membranes. Any lesions on the skin and mucosa membrane allowed to Candida multiply. Now the modern classification of candidiasis clinical forms. Superficial candidiasis of mucous membranes, skin and nails. Chronic granulomatous candidiasis, then visceral candidiasis of various organs. Again, this clinical form is developed due to treatment by antibiotics. Candidiasis of stomach, candidiasis of small intestine are described. When Candida really multiply a lot of in the stomach, in small intestine. Then, of course, in immunocompromised people, systemic or disseminated candidiasis developed, or candida sepsis. You know what does it mean, sepsis? Sepsis means multiplication of microbes in blood. Very often we are talking about sepsis caused by bacteria, but in this case we are talking about multiplication of candida in the blood, candida sepsis. And candida antigen allergic reactions are developed for substances of candida. Clinical features with candidiasis of the mouth. In case of candidiasis of the mouth, a white curdled plaque appears on the mucous membrane, like particles of curd, white particles looking like curd, and they are visible on mucous membrane. In case of urogenital candidiasis, vaginitis, the same particles, white color, are detected on the mucosa, and skin lesions are more common in newborns, looking like small nodules, papules, and pustules. There is a photo. Candidiasis of the oral cavity, mucosa of oral cavity in newborns. Do you see white particles? And it is just acute stage of candidiasis, the initial stage. Firstly, white particles are developed, and then the mucosa becomes red, dry, and irritable, painful, and newborn will refuse from the feeding because it feel pains, and it for newborns, if a newborn will not get a food during one day, then second day, and of course he will be weak, and bacteria will be involved. Bacteria are able to cause bacterial infections at the same time, so it is very dangerous for newborns. And then there is photo of skin candidiasis caused by Candida albicans. Some papules are developed on the skin caused by Candida albicans if the skin is wet, for example. And microbiological diagnosis of candidiasis, and the same, by the way, the same white particles are developed on the mucosa of genital tract if it is vaginitis caused by candida, the same clinical features, the same clinical signs. So microbiological diagnosis. Microscopical test is used in smears from clinical material, pseudomycelium, mycelium, and budding blastospores are detected. But there is a photo. What are they? What? Pseudomycelium. Cells multiply quickly, they stay together, and they are able to develop blastospores. And it is visible. And cultivation. Cultivation is carried out on Saboros agar with subsequent identification, and then they are identified fungi by morphological, cultural, and biochemical properties. If Candida sepsis is suspected, a culture is isolated from the blood culture, hemoculture, and in this case, the blood is inoculated on liquid Saboro medium, not agar. As usual, blood is inoculated always for detection of microbes in liquid medium, and the blood should be taken from vein in a volume from 5 till 10 milliliters, and it should be inoculated on liquid medium 10 times more. So the volume of medium should be 50 or 100 milliliters. It should be cultivated liquid Saburo agar, again special medium for fungi cultivation, and then after we will notice the signs of growth, microbes are re-inoculated on Saburo agar, and we are looking for colonies, and again identification should be done. It is necessary to isolate pure culture, then I will explain you why. Serological method, the aim is to detect antibodies by using of ELISA in the serum of blood of patient. And because of Candida are detected sometimes in healthy people, it is better to use a few samples of the serum of blood taken with intervals 1 week, for example, or 10 days. And if really the patient has candidiasis, it is possible to detect the increasing of the titer of antibodies like PET sera. Those are used in bacteriology and virology. And we have to compare the titer in the first sample of the serum and then in the second. And if the amount of antibodies is increased 4 times and more, means the diagnosis is confirmed by serological method. But if the amount of antibodies still at the same level, means this person had candida before, and now he has maybe some infectious disease, but not candidiasis. As usual, PET sera. Allergic method, and for skin allergic test, Candida allergen is used. It is commercial, it is produced, and it should be, as usual, allergen should be injected into the skin, and then in 3 days from injections, in 72 hours, we should check the result. And if in the place of injection we will see redness and swollenness, papule, it means positive result. So in case of Candida, vaccine is not developed candidiasis, so it is confirmation of diagnosis because of in other cases, if a vaccine is developed, like in case of anthrax, tuberculosis, of course, in vaccinated people, of course, we will see positive result. So it is the interpretation of results should be done carefully. The patient should be asked, was he vaccinated? And maybe he had this disease before, because this allergic reaction is long lasting. So in case of candida, vaccine is not developed, and this positive result means confirmation of diagnosis. Treatment. Treatment is done by, again, specific antifungi preparations. Amphotericin B is often used for treatment of systemic mycosis, for treatment of dermatomycosis, and then flucitazine, fluconazole, itraconazole, Unfortunately, fungi of different species of the genus Candida differ in their sensitivity to antifungal drugs. So I told you that pure culture should be isolated, cultivation should be done, and then after pure culture will be isolated from this person, from this patient. We should do, it is named antibiotic sensitivity test, but really not just antibiotics are checked, different chemicals. We have to realize what medicine this pure culture of fungi is sensitive to. Many chemicals should be used, many medicines should be tested, and finally we should realize which one is the most effective. So different preparations, but anyway, sensitivity test should be done. Otherwise, it will be non-effective, just wasting of money and time. Amphotericin B, for example, for injection. Then there is fluconazole, for example. Then itraconazole, and here is fluconazole preparations, and no specific prevention. Vaccine is not developed. So measures of non-specific prophylaxis, control of asepsis, because of it is possible to develop candidiasis at hospitals due to some invasive procedures, catheterization of veins, blood, by using of sometimes before surgery it is necessary to check is the urine sterile or not, and so blood catheterization is done. So, and in this case we should use just sterile equipment and disposal syringes, plastic syringes there, one-time using, and so on, and safe sex to avoid transmission of candida by sexual contact. Lecture is over. Thank you for attention. Have you any questions?
Thank you very much.
Okay, so if you have no questions, the lecture is over, and you have one more lecture in 2 weeks from now. I will inform you. So
Okay.
no questions? Bye-bye. Goodbye.
Should we do
Goodbye,
the
thank
test
you.
on the
And
portal?
I'm going to remind leaders. Now I will open the test for the lecture on educational portal. Now the time is 6 p.m. So I will open, okay, in 10 minutes, okay? 6:10,
Okay.
or maybe immediately. Then, okay, 6:05. I will open test 6 at 6:05 and 2 hours, 8 till 8 p.m. And then I will close the test. So, and I will check attendance of the lecture due to answering contest, okay, for test. Sure.
Okay.
From 6 p.m. till 8 p.m.
Okay.
Inform all group mates, please.
Sure. Thank you.
Bye. Thank you, Ashraf.
Goodbye. Have a nice evening.
You too.
Goodbye, doctor. Bye-bye.